An acacia tree remains rooted in the savanna for decades, harvesting sunlight, while a cheetah accelerates past it to hunt. On the surface, their lives share almost nothing in common. But at the level of the cell, that contrast disappears. Beneath the skin and the bark, both organisms are constructed from the same foundational units of life. At this scale, the basic machinery is universal. Whether the end result is a predator or a plant, the starting point relies on the same core components. Identifying this shared baseline is the only way to see where these cells eventually diverge to solve the unique problems of survival. Every eukaryotic cell begins with the cell membrane, a flexible, selective boundary that dictates which resources enter the cell and how waste is expelled. This cross-section shows the cytoplasm. It isn't the environment outside the cell. It is the internal, jelly-like fluid where every cellular structure is suspended. At the center of this fluid is the nucleus, a secure, double-membraned vault. Inside, the cell stores its genetic instructions in the form of chromatin, tightly packaged threads of DNA. In the middle of that vault sits the nucleolus, a workshop responsible for assembling ribosomes, the cell's protein-building machines. These universal structures represent the bare minimum for life. From here, specialized adaptations begin to define the divide between plants and animals. A plant cell is designed for a stationary life. Because a plant cannot move to find its food or shelter, its architecture must provide its own defense and energy. In this microscope view of onion peel cells, the thick, rigid borders are cell walls. Made of cellulose, this casing lets a plant grow hundreds of feet tall without a skeleton. Inside, a large central vacuole acts as a water tank. As it fills, turgor pressure pushes outward to keep tissues firm. To power this life, the plant uses chloroplasts. These green organelles function as built-in solar panels, converting sunlight into chemical energy through photosynthesis. Every unique feature here, the rigid wall, the pressurized tank, the solar panels, is a specific structural solution to the limitations of being rooted in one place. Mobile survival requires an entirely different kind of cellular logic. Animal cells are built for flexibility and high-speed energy consumption. This video shows an animal cell's ability to squish and stretch. By evolving without a rigid cell wall, animal cells gain the elasticity required to form intricate, moving tissues like lungs and muscles. Because animal cells have faster metabolisms, they replace large water tanks with smaller vacuoles and dedicated lysosomes, organelles that act as garbage disposals to break down cellular waste. To fuel that movement, animal cells contain a high density of mitochondria. These are the cell's powerhouses, burning fuel to provide the energy needed for every hunt or escape. The animal cell trades physical rigidity for the high-energy flexibility that is required to survive in a world of constant motion. Placing these two organelles side by side reveals their complementary roles in the cellular economy. The chloroplast is responsible for manufacturing food, using light to build sugar molecules. The mitochondrion is responsible for releasing that energy, burning sugar to power the cell's life processes. Plants use both to build and then burn their own fuel. Animals, however, lack chloroplast entirely, meaning they must constantly consume food from their outside environment and rely solely on mitochondria to unlock the energy inside. This microscopic architecture scales up to define the macroscopic world. The cheetah's speed and the tree's height are the direct results of the organelles inside their cells. By manipulating how the vacuole maintains turgor pressure against the cell wall, agricultural scientists develop drought-resistant crops that stay upright and rigid even when the soil is dry. Mapping how mitochondria convert sugar into energy, or where that conversion fails, allows medical researchers to design treatments for human diseases rooted in cellular energy collapse. These microscopic blueprints are highly organized systems where every structure is a specific response to the survival needs of the tree or the cheetah.